Here I have a question. Before we start this topic, this is a word you have heard for the first time in your life. But I have a question, simple question. How many senses we have? Now, you have been hearing, we have 5 senses, and you are saying, my 6th sense is saying something is going to be wrong. Imaginary things, right? You actually have 11 senses. You actually have 11 senses, and the strongest sense of your body is hunger. Hunger is a sense, right? When you are hungry, you don't look for anything else, just for food. And hunger is the thing which can make you do bad things as well, right? Stealing or something else. When someone is hungry or someone's kid is hungry, you'd have seen that, right? So, you see, you know that in Islam, this is that you see, if someone steals something, the punishment is that cut his hand. But I think so, I'm not sure. Yesterday I was, someone was telling that during Hazrat Umar's, reign, when he was the Khalifa, at that time the famine came. I am not sure during Hazrat Umar or Hazrat Usman, I'm not sure, but you can confirm it. And at that time, the punishment of cutting the hands, that was abolished. Why? Because when there is famine, the poor people who will not have anything to eat, they will eventually what? Steal, right? So at that time, I'm not that caliph. He, Radhiyallahu anhu, he abolished that, that punishment at that time for a short period of time when there was the famine. So I'm just giving you this example. Now this gustation, it is the taste, the sense of taste. This is the scientific word for sense of taste. There is another sense of smell, right? The sense of smell, scientifically it is called as olfaction. Sense of smell, scientifically it is called olfaction, and sense of taste, scientifically it is called gustation. Now, the taste happens on your tongue. Now, on your tongue, there is presence of the taste buds. Now, if you have observed your tongue, it has some ridges and furrows, right? It has some areas which are higher, some areas which are deeper. Now those ridges and furrows, they are called as papillae. What are they called? Papillae. Now this is the plural. Singular will be called what? Papilla. Right? Now these are of 3 types. One type looks like this. One type looks like this. And one type looks like this. Now, this type, this is called as fungiform papilla. This one is called as foliform papilla, and this one is called as circumvallate papilla. So there are three types of those ridges and furrows. This is called fungiform papilla, this is called foliiform, this is called circumvallate papilla. Right? Now, in these papilla or in these papillae, the taste buds are present like this. So what is this representing? A taste bud. Right? Now what I am going to do? I am going to enlarge the diagram of one of the taste buds. One of the taste buds. Now if I draw a taste bud, it will appear like this. Now over here, this is the epithelium of tongue. What is this? Epithelium of tongue, right? And this over here, this is a hole or opening. A hole is called what scientifically? A pore. Right? Now this is the taste bud and this opening will be called what? A taste pore. What is it called? Taste pore. On the inside over here, there are receptor cells. What are there? The receptor cells. Let's suppose that this is one receptor cell. This is another receptor cells. Now, just few minutes ago, I discussed with you guys that there are 5 senses, right? There is sweet, salty, sour, bitter, and umami. Right? Now I told you guys that this umami, this is specific for the amino acid glutamine. Or we call it as glutamate as well. Do you remember this or not? Right? Now where have you learned about this glutamate before? Can anyone tell me the example? Excellent, well done. Hemoglobin, 6th amino acid of alpha or beta chain? Beta chain. Should be glutamate, but what happens to it? It is exchanged with valine in sickle cell anemia. Remember? Now, which food you will eat that will have glutamate in it? Imagine. Any meat. All the meats, they are what? Proteins. When those proteins are digested, they will have glutamate definitely in it. So it means that the umami is actually the taste bud that gives you the taste for meat, for the amino acids. Got it? So this is sweet. It can detect glucose or sucrose, right? Salty, it will be detecting sodium and chloride or the other ions, right? Sour, what will be the sour detecting? Acid, well done. And bitter, these are the chemicals like capsaicin chemical, which is the chemicals present in the, like you see in different spices, right? In chilies, we can say that, right? They are bitter. So over here in the taste bud, there will be taste receptors for all 5 type of taste. Now, from here, now these cells which I just drew, these cells are actually taste receptor cells. These cells are what? Taste receptor cells. And these taste receptor cells, all of them, they have cytoplasmic extensions over here. They all have what? Cytoplasmic extensions over here. Now, these cytoplasmic extensions, these are called as microvilli. What are they called? Microvilli, right? And from here, what happens? That the nerves or the axons of the sensory neuron they go towards the stem of the brain, towards the base of the brain, right? So remember that the message first of all goes to the, because you see the tongue is here and stem of the brain is over here. So the message goes first to the stem of the brain, then you see there are different centers in the brain for detecting different tastes. So according to the chemical, the taste signal will go there. We are not going into that detail. You will learn that in medical school. Now, so you guys have learned the structure of a taste bud, right? This is the epithelium of the tongue. There is a hole in the epithelium of the tongue, right? In the papilla. That hole will be called taste pore. Inside there are taste receptor cells. Maybe this taste receptor cell is for sweet. This is for the sour. This is for the bitter. This is for the umami. This is for the salty, right? So there are all types of taste receptor cells over there. And these taste receptor cells, they have axons or dendrites attached with them. Got it? Which will take the message to the brain. Now question, next step. How the taste will be detected. How the nerve impulse will be generated. This is the question, main question. Okay, we put salt on the tongue, we can taste it. But how did we feel it? How it created an action potential? How it created depolarization? That is what we are going to discuss. For that purpose, what I am going to do, I am going to draw a taste receptor cell. Let's suppose, what is this? This is a taste receptor cell, and this over here is a nerve ending. This over here, this is the taste receptor cell. And you guys know that the taste receptor cell, it did have what? Microvilli on it. It's a cell, so definitely it will have what? Nucleus as well, right? Now, I will give you the example of the others as well. But first we are going to discuss the example which is in your book. But there must be in your mind a question how the sweet is detected. In your book they have only mentioned salt one. But I will tell you how glucose will be detected or how it will cause the action potential, right? First, let's talk about the structure of this taste receptor cell. Let me mention, this is what? This is a taste receptor cell. Now, in the taste receptor cell, obviously there are sodium ion channels, right? There are potassium ion channels. But over here, there are calcium ion channels are also present. What are these? Calcium ion channels. Now what I am going to teach you today, I will ask you in the next topic when we learn about the synapse, inshallah. Right? Tomorrow we'll discuss the synapse. So I'll ask you that thing. Now, I have a question. You guys have learned that in O levels. At the axon nerve ending, there are small vesicles that have neurotransmitters in them. What are those vesicles called? Who remembers? Sorry? In the axon nerve endings, there are small vesicles that contain neurotransmitters in them. What are those vesicles called? Close. Speak louder. Excellent. They are called synaptic vesicles. Right? So over here, in this taste receptor cells, there are synaptic vesicles are present. Vesicles that contain neurotransmitters. So what are these? These are synaptic vesicles. You guys have learned in the synapse, the nerve ending never touch the other nerve ending. So this nerve ending will not be touching the receptor cell. There will be gap between them, right? But on the nerve ending, there are present what? Neurotransmitter receptors. So what are these? These are neurotransmitter receptor. Now obviously this will be a dendrite nerve ending. Obviously this is what? A dendrite nerve ending of sensory neuron. Obviously what is it? A dendrite nerve ending of sensory neuron, right? Now, so remember now that this is a taste receptor cell. These are microvilli, it has nucleus, it has calcium ion channels, right? Obviously, it has sodium ion channels as well, right? What is this? Sodium ion channel. Then, it has synaptic vesicles which contain neurotransmitters. Now, there are different types of neurotransmitters, right? There are, now, you know, we learned in the beginning of this chapter, there is peripheral nervous system and there is central nervous system. The neurotransmitter present in the peripheral nervous system is only one. Its name, I'm going to tell you right now. Next topic I will ask you. Acetylcholine. I'm writing over here. This is the neurotransmitter of peripheral nervous system. Means that all around the body, wherever there are connections are there, this acetylcholine will be there. It means that here, these synaptic vesicles will have what? Acetylcholine. And these receptors will be for what? Acetylcholine receptors. Understand that? While in your brain, there are different types of receptors. Dopamine maybe you have heard, is one of them. Right? Endorphins, maybe you have heard, are one of them. Right? Now what happens? Here we are only, first of all, going to talk about what? Salty. Now, when you put salt on your tongue or you drink something which is salty, or you eat something which is salty, in that the salt will be divided into what? Sodium and chloride ions, right? It's obvious. What will happen? That these sodium ions, they will pass from the outside to the inside. Sodium ion channels. Obviously, from higher concentration to lower concentration. When sodium ions will move inside, the charges, depolarization will take place or not? That depolarization will open these calcium ion channels. That will open calcium ion channels, and because of that, what will happen? The calcium will start moving from outside to the inside.
So can you repeat how it works?
Yes. Sodium, you see, you ate something which is salty. It means it has sodium ions in it. Those sodium ions will move from higher concentration to lower concentration, from outside of the cell to the inside of the cell. That will cause the depolarization, right? That will change the shift in the charges, as we learned yesterday. That shift in charges will open calcium ion channels. And because of that, when the calcium ion channels, they will open calcium ions, they will move inward. So calcium ions, they have come inside. Now, this thing is not mentioned in your book, but without this you will not understand. So I'm going to tell you. These synaptic vesicles are not present freely in the cytoplasm. They are not present freely in the cytoplasm. They are bind to the membrane with the help of special proteins. With the help of special proteins, and these proteins are called snare proteins or snare complex. Yes, sir.
Does the chlorine move with the sodium?
No.
Why is chlorine there?
See, it caused the depolarization. Remember, these are voltage gated channels. They open at a certain voltage, right? So calcium, you see, when the sodium came inside, depolarization happened, and that depolarization led to the opening of the calcium ion channels. Understand? Change in charges led to the opening of the calcium ion channels, right? And the calcium, when it moved inside, now we are going to discuss the function of calcium, how it helps. So what I am saying, that these vesicles are not present freely in the cytoplasm. They are bind to the cell membrane with what? Snare proteins, right? Now what will happen? The calcium ions, what they do? They bind to snare proteins. These are also called as snare complex. Now in response, these snare proteins, they will contract. In that response, the snare protein, what will happen? They will contract. When the calcium ions bind to the snare protein, they will cause contraction of the snare protein. Snare proteins will contract. Now what will happen if the contraction takes place? The vesicle comes near to the membrane. And when the vesicle comes near to the membrane, one process will happen. What is that? Exocytosis. Excellent. When the exocytosis will take place, what will happen? The neurotransmitters will be released where? In the gap. And those neurotransmitters, what they will do? They will bind to what? To the receptors on the dendrite nerve ending. They will bind to the receptors on the dendrite nerve ending. When they will bind to the receptors on the dendrite nerve ending, which channel should open? For the transmission of nerve impulse, which channel should open? We learned yesterday. Sodium ion channels. Well done. So what will happen? Over here, sodium ion channels, they open and the nerve impulse is transmitted. right? So you just put the,
So the voltage.
you just a second. You just put salt on your tongue and right away you feel it. And it's so complicated. Right? Yes. What is your question?
So this Na plus channel is the voltage gain?
Obviously. Right? Now, listen again, very quickly, I'm going to repeat it. This receptor cell has microvilli, has sodium ion channels, have calcium ion channels, has synaptic vesicles attached with snare complex to the cell membrane. Right? When the sodium ion concentration increase, obviously it will increase when you will put something salty on your tongue. Sodium ions will move from outside to inside. Higher concentration to lower concentration. They will cause depolarization, which will lead to opening of what? Calcium ion channels. Calcium will go inside and will bind to what? Snare protein. Because of that, snare protein will contract. As it is shortening, the vesicle will come near to the membrane, and what will happen? Exocytosis. Because of exocytosis, the neurotransmitters will be released where? In the gap. And then these neurotransmitters, they will bind to the receptors. And that when they will bind to receptor, an action potential will be generated that will open the sodium ion voltage gated channels. And the nerve impulse will travel. It will go where? To the brain stem and then to its proper brain center. Understood? So this was how the salt is detected. Question is how, let's suppose glucose will be detected. In the sweet, there is glucose there or there is sucrose there. The sodium was simply going from the sodium ion channels. There are no channels for the glucose or sucrose over here. There are no channels for glucose or sucrose over here. Now what can happen? Anyone give an idea? You are drinking glucose, not the starch. No. No. There are receptors present on the surface of the receptor cell. There will be what? Excellent answer. Cell signaling. Receptor present on the surface of the receptor cell. Glucose will come and bind to it. What will be produced then? CAMP. G protein will be activated, all that process. We have learned that in AS. I'm not going into that detail, right? And when you guys came into the lab, that exactly the same cycle was drawn on the board, right? So what will happen? CAMP will be produced as secondary messenger. Remember that or not? And that secondary messenger will lead to the opening of the sodium ion channels. And then what will happen? Then sodium ion channels will come inside. Sodium will come inside. That will lead to the opening of calcium ion gated channels. And eventually the reaction will take. This whole process will take place. Right? So for sodium it is simple. But for the other like sweet, sour, bitter, umami, there will be receptors on the receptor cells. Right? Which will, that will be like secondary messenger and all those processes will take place. Understood? So this is how the taste is detected. This is how gustation works. Now this is not in your syllabus, but just for your information, a little bit about olfaction. Now, this angle. Now this over here is your nose, right? Now, in the nose, there must be some receptor cells that will be detecting the smell. The smell is because of the chemicals in the air. Now, if I put some perfume, that perfume, you see, it is releasing the chemicals from higher concentration to lower concentration, from my clothes to the air. That's why you can smell. That is the reason that eventually the smell fades away. Because the molecules, they are moving away from higher concentration to low. You see, if you put the perfume in the morning, by evening time, enough smell will not be there. There are two reasons for that. One, that the molecules of the perfume are going away from higher concentration to lower. Second, you are becoming used to of the smell. After some time you say that I don't smell anything. Right? We'll talk about that. That is also a very interesting factor actually. That is related to threshold. I have to discuss this, okay? Now, over here, your brain is present. Right? Your brain is not floating. Your brain must be present on some bone present over here. So over here there is a bone on which the brain is resting, but this bone has pores in it. This is called as cribriform plate. From here, from the anterior end of your brain, the receptor cells are coming down into your nose. Now, if there are 10,000 types of smells, there will 10,000 types of receptors over there. Now, if you go and you smell biryani, the biryani smell is because of the chemicals in the air coming from the biryani. Different spices over there. Hyderabadi biryani, different taste, different smell. Right? Sindhi biryani, different smell. Right? Karachi nihari, different smell. So because they have put different spices in them and they are making it aromatic, right? Releasing different odor from that. So different, you see these nerve cells, they become excited over there. Got it? So this is what you see there are a few examples of what? Of the senses.